Welcome everybody to the LINC Showcase. I'd like to welcome everybody who has come into the auditorium and many people who are actually online. Not surprisingly, things are a little bit tricky for a number of people related to illness and things at the moment. So I'd like to begin this afternoon with acknowledging the traditional custodians of country throughout Australia. We recognise their continuing connection to land, waters and community. I pay my respects to them and their cultures and to elders past and present. So this is our second LINC Showcase, and I again extend my welcome on behalf of our CEO Scott Bell, who hopefully is online and watching as well. So as you would recall, or you may be new to this program, LINC is about, as the name suggests, linking clinicians and researchers to come together to form collaborations to address healthcare challenges and otherwise. So the showcase and the format of the showcase is an opportunity for both researchers and clinicians to present their pitches, and that might be the research conundrum that they have that they would like clinical input from, or it might be a clinician who has a question that they need help to answer. And so this afternoon we'll have 13 research pitches and some Q&A in between. We have some expert panel members, and I'd like to thank Dr. Tamsin Terry and Dr. Erin Raymond for coming along today, and they'll be helping us to ask some questions. I will be absolutely ruthless with the timing today. So you have your 3 minutes, and we have 3 minutes for question, and that will be it. And so please don't take any offence to me cutting you off or otherwise. This is the nature of pitching in this environment, and all of the speakers today have been well informed of and certainly are prepared for their pitches in this way. So I will introduce everyone individually prior to their pitch, and then, as I say, we'll have some Q&A afterwards. So I'd just also like to thank our collaborators in the LINC program, and that's Marta Research and Metro South, and to remind you all, and I'll give you another reminder of this at the end, that of course the LINC grant opportunity will open shortly, and we'll give you some more detail in regards to this, but we would encourage you all to use today as a springboard to think about the grant opportunity that results. The showcase today is for everyone, but I would also encourage you to think that for the collaborative grants, the principal investigators, both the clinician as well as the researcher, need to be early and mid-career researchers. And that is absolutely strategic on our behalf, and we don't shy away from the importance of supporting our early and mid-career researchers. So without further ado, we will begin. We do have a couple of presenters who are presenting online due to a number of reasons, so just bear with us while we begin. So our first speaker presenter today is Dr. Taylon Gagency. He'll be presenting "Do either genetics or the severity of cancer-induced inflammation predict symptom response to medical cannabis in adults with advanced cancer?"
Well, look, thanks so much for having me over Zoom. My name is Taylon Gagency. I'm one of the palliative care research fellow at Diamantina. And I guess, you know, as the title suggests, we're doing a lot of research with medical cannabis at the moment. And one of the things about medical cannabis research, I suppose, is that it's really hard to, A, you know, find which patients benefit, if any, and B, for those that do say they benefit, why that is. And I think, you know, to an extent, clinicians have taken a bit of a potluck approach to this and just tried to identify certain subsets of patients which may respond. But there is some basic science literature to suggest that maybe we can be a bit more refined, I guess, with how we sort of pick these responders. And if we just go to that next slide there, if we can, which I'll assume is done. Look, I think, you know, and I've actually had someone contact me already, which I was really chuffed by. So thanks for that, Manira. I look forward to working with you in the future. One of the things that's in the literature is that perhaps it is the degree of inflammation in some of the patients which dictates their symptom response to cancer. And to be really clear, we're talking about symptoms here. We're not talking about treating cancer or anything like that. I certainly don't think that there's a role for cannabis from that point of view. And I guess different cancers have a different degree of inflammation associated with them, and that's too big a topic to get into at the moment. Nevertheless, though, we have 3 RCTs going at the moment, or one just finished, another 2 that are funded to complete, and probably about, I guess, 450 patients in total with samples that we can draw from them. And we want to look at whether the people whose symptoms improve, whether their degree of inflammation as measured by some cytokines is different to those who don't respond. The other candidate, I suppose, for mechanisms is whether there is some sort of, and we sort of tend to postulate this a lot as clinicians, some form of genomic variation which predicts response. And while sometimes that's a bit of a fishing expedition, there is some literature in the psychiatric literature especially, which might suggest that it could be something to look at for cannabis. So just the last slide now. So look, I mean, just to summarize, what we're looking for is either someone who can measure inflammatory markers and also be able to communicate those findings to an audience primarily of clinicians, or similarly, someone who can look at candidate genes for us. And I know sometimes those are really hard to present in a way that makes sense to clinicians, but just that communication skill would be vital too. I think that'd be pretty close to 3 minutes, so I'll wrap it up there. If you have any other ideas for collaboration with palliative care clinicians that are, very active in research, then I'd be very happy to hear from you, whether it's this topic or something else. Thanks very much.
Excellent, Taylon. That was 3 minutes and 4 seconds. So you're a winner. I'm going to open up for questions. I have one while people are letting me know. What samples have you collected so far over this cohort? So what do you have for the purposes of the scientists to do the cytokine studies and otherwise?
Yeah, that's a really good question. Look, we do have one collaborator already, I suppose, who's going to look at the current samples. So excluding that, the current ones, mostly EDTA tubes. And we've got them frozen and we sort of batch them and we, we'll just look at them, I guess, at the end. We have funding for the current one. We could apportion some funding for the next 2 trials as well to look at cytokines, but it would sort of depend on, I guess, who's interested and what we can do.
So you could prospectively collect samples?
Yeah, for sure. And in fact, our 3rd trial, which is looking at sort of THC CBD 1 to 20, which might not make any sense to anyone, but it's just a different ratio. That hasn't started recruiting yet, and so we could amend the ethics to include that specifically if someone had something in particular they wanted to look at.
Thank you. Other questions? Yes, Rick.
With the inflammatory cytokines, I guess you've got the genomics, which is obviously kind of set in place, but what's the experimental design around the cytokines? Do you just look after acutely after treatment or over long term, etc.?
Yeah, really good question. Look, first thing I'll say is it's a pilot study within the main study, so it's not a primary outcome and it's not powered to say anything dramatic. It's just an exploratory thing. The second thing I would say is that no, it's not related to treatment. The patients are, I guess the primary outcome of the trial is to see whether medical cannabis helps with something called total symptom distress, which is just a, overall symptom score. And so being a pragmatic study, I guess we take all comers with advanced cancer, so long as they're not starting a new or experimental treatment within 7 days of initiating the medical cannabis. So it's quite a heterogeneous group of patients. I think ultimately, it's not, as you sort of imply, it's not powered to make any firm conclusions about inflammatory markers, but perhaps if there is a bit of a signal with a pilot study, that might be something we can look at with larger numbers in the future.
Excellent. Thank you, Taylon. Very much appreciated, and thanks for being able to come online and present today.
Oh, no, thanks for your time.
Thank you. We're going to move on to our next presenter. And that is Dr. Camille Rae, who's an ARC-DECRA Fellow with Bada Research, and she's going to speak on understanding the multifaceted roles of natural killer cell populations in cancer. Thanks.
Good evening, everyone. I'm Camille Rae, and I'm an immunologist. So my work aims to better understand immune responses to cancer with the end goal of using patients' own immune system as a treatment. In Australia, there is over 1 million people whose life has been affected by cancer. And these patients often go through very toxic treatments such as chemotherapies that cost the Australian health system over $6 billion a year. So we need alternative therapeutic strategies. My research focuses on a type of immune cells called natural killer cells, or NK cells. NK cells circulate through the blood and patrol our body to kill dangerous cells, such as cancer cells. NK cells are a very attractive therapeutic target because they act rapidly and they are safe. They can be harvested from healthy donors and transferred into patients without any toxicity. One important fact about NK cells is that they do not all look the same. But even more importantly, they do not all respond the same way. It's a bit like people. When facing a danger, some may attack, others might call for help, but some people might just freeze and pass out. Well, some NK cells might kill the cancer cells, other NK cells might alert the whole immune system, but some NK cells might have an unproductive response that will allow the cancer to grow. So we have developed tools to analyze NK cell diversity from cellular suspension. And with this technique, we were able to identify novel NK cell populations in acute myeloid leukemia samples. So with this tool, we want to test our hypothesis that not all NK cells are good. And we want to define the good guys and the bad guys. So for this, we look at the proportions of different NK cell populations and assess any correlation with disease outcome. This will allow us to identify new biomarkers by linking one specific NK cell population with a given prognosis. And this will also help us to refine NK cell therapies so the right NK cell population could be targeted to the right patient. So this approach could be applied to any cancer type. And I'm looking for oncologists who are willing to work with me to further develop this project idea so we could better understand NK cells and bring NK cell drugs to patients. Thank you.
Thank you, Camille. That was excellent and just a couple of seconds under 3 minutes. Well done. Do we have some questions? Yes.
So you've mentioned that you're interested in all cancer types. Do you have, one or two that you might think might be a preference, might be good ones to start with? And in terms of access to samples, how many samples are you looking at in terms of that sort of access, just to give people an idea?
So my main interest is blood cancers. But this could be really broaden to any cancer. I'm really interested in blood cancers. And also NK cells are particularly efficient against blood cancers. They have difficulties to infiltrate solid tumors, but they are really good against metastasis. So in solid cancers, they could have a good function against metastasis. But blood cancers, NK cells are really good. And in terms of numbers, I think I will have to speak with a statistician. And this is something to refine when refining the project, talking with the clinician, what is feasible, depending on what's the access to sample, I really need the clinical expertise and also the statistical expertise around that to refine the project.
Excellent, thank you. Any other questions for Camille? Yes, please.
Given the interplay between chemotherapeutics or immune therapeutics and the immune system, but also the challenge with later stage patients being heavily treated, are you going to really looking for samples from untreated patients, or are you looking through the whole treatment continuum?
Again, that's something that we could refine, because we can look at diagnosis and see, okay, that's where the NK cell populations are, just at diagnosis, and that's how the patient responds to any type of treatment, and that's how the patient did. Or we can also look after treatment and say, okay, this is the state of NK cell populations in responders to the treatment, and this is the state of NK cell population in non-responders, which would be nice to be able to link it to the pre-treatment sample, but if we can't, this will still be useful information.
Excellent, thank you. So any haematologists, budding or otherwise out there, early career, mid-career haematologists, or anyone that could link Camille up would be excellent. I'm very pleased to introduce Dr. Greg Kear, who's a respiratory physician in the Department of Respiratory and Sleep Medicine here at the PA, and he's going to talk on predictive biomarkers in connective tissue disease associated interstitial lung disease. There we go, Greg.
Thanks, Helen, and thanks, organisers. So connective tissue disease is an umbrella term that encompasses lots of individual diseases, such as systemic sclerosis, rheumatoid, and some of those other conditions listed at the top of the slide there. While they mostly affect the muscles and joints and skin, lung complications are fairly common and potentially life-threatening. One of the most serious is interstitial lung disease, or ILD, which is characterised by inflammation and fibrosis affecting the lung tissue. Because of the potential seriousness of ILD, accurate assessment of CTD-associated ILD severity and disease trajectory is important when prognosticating and deciding what treatment is best for which patient. To help with this, there are a number of staging systems that have been developed over the years to help predict CTD ILD disease behaviour, and these often incorporate findings from CT scans, lung function, and various clinical parameters. So on the right of that screen are a couple of CT scans from two separate patients, the top one with relatively limited ILD, the bottom one with more extensive ILD, and that's how we would grade those based on current staging systems. However, these staging systems have a number of limitations, as highlighted by the case of this young lady whose CT scan is at the top of the screen. So if we use the staging systems currently most widely employed for scleroderma, we would classify her as limited stage ILD associated with her scleroderma, and if we look at those survival curves at the top right of the screen, the limited stage survival curve would suggest she'd have a very good prognosis out to 10-15 years. However, that turned out not to be the case, and she passed away from complications for her disease about 3 years after diagnosis. So because of the limitations of the staging systems we currently use in these diseases, identification and validation of molecular biomarkers is increasingly important. We know in other ILDs, most notably IPF, molecular biomarkers have shown prognostic utility, but the same results haven't really been expanded into the field of connective tissue disease-associated ILD, and that's the purpose of why I'm here today. So we have designed this study called the PREDICT ILD study with the aim of evaluating the prognostic utility of serial biomarker measurement in predicting CTD ILD severity and disease trajectory compared with the tools that we conventionally use in the clinic, so things like clinical assessment, CT scan, and lung function. So we'll be recruiting patients prospectively through clinic and collecting blood at baseline, 6 months, 12 months, and annually out to 3 years. There is a whole array of potential molecular biomarkers that we'll be measuring that seem to change all the time, and that's part of the reason for being here today and looking for expertise in people in some of the basic science of this that I don't have myself. So whether they're markers of alveolar epithelial cell damage, extracellular matrix protein, remodelling, or immune system dysregulation, that's what we're looking to measure in these patients. And the outcome of the study will be looking at biomarker concentration both at baseline and then serial over time, which has not yet been done, which we will then compare against these kind of routine measures of clinical disease severity and progression, like I mentioned, CT scan, lung function, and patient-reported outcomes. Thank you, that is all. I hope I made time.
You were only about 11 seconds over, so I'll let that one go.
Thanks.
So, questions from the audience? Yes, please.
How many patients are you planning on recruiting?
So this will be a pilot study. We're looking for an initial 50 patients. Then we'll do a more formal power analysis, and it's more than likely going to be a collaborative effort with other centres in Australia and overseas.
So how long do you think you'll take to get those 50 on board?
12, 18 months. But then they'll be followed up for out to 3 years.
Greg, is there any preliminary immunological data or otherwise that would help your basic science colleagues sort of plan? I mean, you've mentioned a few up there.
Some of these assays are measured by sort of commercially available ELISA kits. Increasingly, and this is kind of the question that I don't know the answer to, there seems to be these, certainly with plasma protein analysis, these multiplex high throughput analyzers, which seems to be where this field is going, and sort of having access or expertise to that's going to be important. And I think when we get to measuring these biomarkers, that's probably where we're going to be heading.
Yes, Rick, please.
Just already, I mean, you mentioned the other, maybe it's a more severe idiopathic disease, that there are some established markers. How much is already known about your particular interstitial lung disease? Are there already some markers in the blood?
There is some precedent in this area, and there is a little bit of overlap with idiopathic pulmonary fibrosis.
Right.
But the studies that are then rolled into connective tissue disease ILD have been very small, often retrospective, and haven't followed patients with serial measurement to gauge change in biomarker over time.
Right.
So not much in this field, in short.
You do think you could distinguish the two with specific markers? There'd be specific markers different between the two?
There may well be, given they're pretty distinct diseases, but I'm less interested in it as a diagnostic marker, which I think we already have good diagnostic tools. It's more of a prognostic marker.
Thank you.
Thank you, Greg.
Thank you.
Excellent. Thank you, Greg. So researchers out there, you're going to come to a collaboration with an excellent potential biobank. Our next presenter is Dr. Arutha Khal, who's a Senior Research Fellow and a Group Leader at UQDI, and he's going to talk to us about understanding resistance to immunotherapy, a multi-omic approach. Thank you.
Hi, everyone. Today I'll be talking to you about sort of a multi-omics approach to delineate signatures of IO resistance and response. I'll flag that image in the middle that's sort of flicking through there. It's a really unique case, but shows how we can apply this really rapidly. So IO is really effective in a number of tumor types. This work's really been led in melanoma, triple-negative breast, bladder, head and neck, non-small cell lung cancer. One of the challenges is really identifying those predictive biomarkers of checkpoint inhibitor therapy. About 15 to 40% of patients respond, depending on tumor stream there. This paper came out in Nature last year, which is a commentary, but it really showed you why being able to profile the tissue, being able to get that imaging data, the genomic data, the proteomic data simultaneously off that, and being able to visualize that is really powerful, and how that's really moving the landscape forward. I'll talk about this really that unique case that we've highly profiled now, but we're able to run this en masse now within the lab, and we've got the workflow. So this is what a standard pathologist sees down a microscope. And what we're able to do is we're able to look at so this is a dual color H&E stain. What we're able to do is we're able to look at about 100 different markers on this tissue, 100 and more markers on this piece of tissue. And this is from a head and neck patient that had a partial response to IO therapy, died after 14 months. And we had the patient's biopsy sample, and we were trying to interrogate it. And I'll talk to a bit of that work that went through it. So we're able to generate these pretty multiplex IF images, but we're also able to develop a cellular atlas. When you're able to plex above 50 colors, and this is at 103 plex, we're able to get all this tissue architecture details coming through. So we can see very clearly that there are these tertiary lymphoid structures. Those are germinal centers in the middle, but tertiary lymphoid structures, which are infiltrating this bit of the tumor here. The bit of the tumor at the top right, but it's not in the other bits of the tumor. When we look at Ki67 co-expression with a few other markers, we can see we can break this tumor up into multiple different tumors, so tumor region 4, 3, 2, and 1. And then we can see that when we go back here, we can see that these tertiary lymphoid structures are associated with an immune-responsive microenvironment. There's a lot of literature that's come out in this space which showed that B-cell infiltration into the tumor sustains an immune-responsive microenvironment, but also the structure of these TLSs is key. So when we break this tissue down in situ into about 900,000 single cells, we can break that tumor up to 4 different regions. All the M1 macrophages are localizing to that bit in the yellow. The M2 macrophages are localizing to the other bit. So M1s are immune responsive. The M2s are immune suppressive. So on sort of 2 different dimensions, the lack of tertiary lymphoid structures throughout the tissue the M1 macrophages being around where the TLSs are, and then M2 phenotype being everywhere else, shows you why this patient potentially had a partial response to IO therapy. We can also start to look at other markers, so metabolic data on these tissues. So this is now looking at BAX, which is a pro-apoptotic marker, which is all in that bit at the top. So that's all dying at the top in the green, and G6PD, which is a marker for metabolic activity, and tumor region 3 is highly metabolically active. So you can now start to understand on multiple dimensions, T-cell infiltrate, M1, M2 max, metabolic data, why this patient potentially had a partial response to IO therapy. For this, we use a team science approach using spatial transcriptomics, spatial proteomics, and we sort of crunch these two together using a computational approach that's now been developed in the lab. Thank you.
Thank you, Arutha. Just 20 seconds over, but tell us what collaborators are you looking for?
So we're looking for prospective studies at the moment where we can, so we're doing sort of large studies retrospectively, but to validate these signatures, we do really need the access to prospective samples. That's been key in head and neck and lung cancer. So we are looking for collaborators that have access to those cohorts.
I'm just curious, have you done any immunoprofiling on the original biopsy site versus a metastasis? And does that give you any clues?
That's a good question, yeah. We published a paper earlier this year looking at primary versus nodal mets in head and neck, and you can start to see clear differences between the two when you use this approach. So the challenge is in getting the biopsy from the metastatic site. For lung cancers, it's hard. Head and necks, some cases we're able to get them. But yeah, primary versus nodal and also longitudinal prospectively is going to be key to really validating these signatures.
Okay. Other questions? Yes.
Is this something that you need whole tissue for resection for? If you don't need all that too?
Just FFP, any archival tissue. We've worked off FNAs, we've worked off cytology specimens, tissues that are 10, 20 years old. We're about to run a project on Spanish flu, and that's 100-year-old tissue. So, yeah.
Any other questions? Otherwise, we'll thank Arutha. Thank you very much. So our next presenter is Professor Derek Richard, who's the Scientific Director of the Cancer and Ageing Research Program from QUT here at TRI, and he's going to talk to us on Cancer and Ageing Research Program, understanding the link, well done, got it in there, between age, genetic damage and disease. Thanks, Derek.
All right, so I'll tell you about us as a group rather than about an individual project, because we have many ECRs and MCRs within the Cancer and Ageing Research Program who are interested in working with the clinical interface. So effectively, CARP is a group of researchers, about 60 of us, or 64, and we really want to understand why genetic damage drives aging and drives diseases of aging. If you think of the number one risk factor for cancer, it's age. For dementia, obviously, it's age, osteoporosis. So we're trying to understand what that link is between age and how we repair our genetic damage. So as a group, we're pretty large. As I say, we formed ourselves more like a biotech company. Now we do things. So we have what we call discovery biologists. They're discovering new science, new DNA repair proteins, trying to understand pathways better. And then they lead on to, so they're our group leaders. Our team leaders then move along the translational pipeline. So we have a $7.5 million drug discovery center in our lab on level 6 of TRI. So we can screen up to 80,000 compounds. We can get your libraries, etc., whatever you want to do, and how little you want to be involved or how much you want to be involved in that. Our computational team, they can screen up to 3 billion compounds, and they do in silico and molecular dynamic screening to get that. But ultimately, once the drug is found, we can move through to the preclinical team, and they'll do the models that you understand, and then they can eventually move on to the clinical team, and ultimately towards developing a drug or a therapy of some kind. So as a lab, we've 3 spin-out companies have come out of our lab, so we've done pretty well. That's since moving to TRI in 2013. So we have a strong partnership with some clinicians already, but we want to build that partnership with clinical interface. We have interest, obviously, in respiratory diseases, COPD, lung cancer, etc. Anything where the DNA damage system is either being pressurized by the disease state or that mutations are driving that disease in the first place. So we have a strong interest in those areas. We have obviously cancer, we have an osteoporosis team working up there, we're moving into dementia. And so that's anything where the DNA damage system has been targeted in some way. So basically, I call out to any clinician in any area where they think that the DNA damage response may be involved, which is, I'm going to argue, is most aging diseases. And we have interest in molecular gerontology, etc. So broad range of interest. If you want to see our drug discovery unit, just come up to level 6 and we're there. Thank you.
Excellent, Eric, and perfectly to time. Questions? So I guess, Eric, any chronic disease is open for, your research groups purvey, because, I just think about, the largest chronic diseases are all potentially or most often related to age. Correct?
Yes, yeah.
So you really got a broad audience out there.
Yeah, we have a very broad audience, probably about. So I guess the thing that pulls us to where we are is the fact that, I'm Scottish, if you didn't guess. So if you look at 1980s man in Scotland, you retire at the age of 65 and be dead by the age of 68 on average. But we're really getting the clinicians who are really good and the scientists, really getting people to live longer, but only through medical intervention. So if you look at health span, we still get cancer at the same stage and all these other diseases. We just can treat them. So we're looking at why we get these diseases and what we can do about them.
Excellent. Other questions?
So Derek, in terms of the clinical collaborators, so what's an ideal collaborator for you? Like, how much do they have to get involved? Are you looking at people to, hypothesize around why things might happen? Are you looking for sample collection? What might it look like?
Yeah, look, it comes in all sizes. We don't have a robust strategy for it. We have some clinicians who have come and done MPhils in our lab. That's quite a common interface, but you can have other people where they just want to ask a particular medical question and we can see what we can do there. When it comes to drug discovery, you can either come with an idea to us and we'll help you develop it, or you can come with a target, an assay developed, and we can just then run it through the screen, so much easier. So it's basically up to the time that the clinician can spare and it's fit with what we do in the lab.
Excellent, thank you, Derek.
Yep, thank you.
Okay, so that's really a collaboration that's prime for a number of our clinicians. So I'd like to introduce Dr. Sophie Bergo, who's a research fellow with UQDI, and Sophie is going to talk to us about microbial sequencing in liquid biopsy for early diagnosis of spondyloarthropathies. Thanks, Sophie.
Hello everyone, and I'm an immunologist here at UQDI on level 6, and my research project is about the large family of arthritic conditions such as spondyloarthritis or SPA. It's a progressive autoimmune disease that affects about 2% of the global population and is targeting the joint. So you can have inflammation of the joint of the neck, the pelvis, the hip, the hand, the knee. It's very large representation, and it's giving pain and stiffness to those patients. Up to 70% of those patients also have gut symptoms that can develop into inflammatory bowel disease in 10% of them, also known as Crohn's disease or ulcerative colitis. Some patients can also develop skin inflammation such as psoriasis or eye inflammation just such as uveitis. In 2015, the overall cost, the health cost of these in Australia was $5.5 billion, plus an extra welfare cost of $1.1 billion. This number is about to rise by 50% in 2030. Today, there is no cure. We can only relieve the pain of the patient. We can give some treatment regimens to this patient that will halt the worsening of the condition, but some of these patients can also become resistant to these drugs. So we need to find something else. It really affects the quality of life of those patients, and you must know that arthritis is the second most common cause of disability and early retirement due to illness in Australia. So we need new options for these pathologies. The gut microbiome is composed of trillions of bacteria that are living there in equilibrium with the host. But the gut microbiome has also been associated with pathology in which we have an unbalanced microbiome that is also caused dysbiosis. And SPA patients, spondyloarthritis patients, do have an overgrowth of bad bacteria over good bacteria in the gut. My preclinical work on a mouse model of spondyloarthropathy has shown that gut-derived bacteria can actually cause the progression of the disease in the joint, and we did find some bacterial motifs in the inflamed ankle. But we don't know how they get from the gut to the ankle, and we think it's through the blood. There is actually a healthy blood microbiome. Some people can have bacteria detected in the blood, and also some patients with gut symptoms have shown that their gut microbiome differs from the healthy gut microbiome. So there is a link between the gut microbiome and the blood microbiome. Septic patients can be diagnosed towards targeting bacteria in the gut, and this can allow them to get an appropriate treatment for those patients, so that we can use the blood as a liquid biopsy for diagnostics. So in this study, I want to be able to use samples from SPA patients and compare the microbiome between the stool, which would be a mirror for the gut, the blood, and the joint of this patient. If we find gut-derived bacteria in the blood and in the joint of this patient, then it strongly suggests that you have circulating bacteria, bacterial motif or DNA that goes from the gut to the joint through the blood. And then the blood becomes then, the blood is a new tool to be able to detect early those patients with ongoing psoriatic arthritis. So this project also has the potential, of course, to be used in other autoimmune diseases that we have gut symptoms. We could triage patients with gut symptoms and then predict the outcome and then be able to offer them adapted treatment option. To move this forward, I'm actively looking for collaborators with clinical expertise and understanding of the pathology involved in this project. Please contact me if you are interested. In the meantime, eat well, drink wisely, exercise, do not stress, and keep your gut healthy. Thank you.
Thank you, Anne-Sophie. I won't ask a question because, as you know, I have a vested interest in this area. So any questions from the audience? Yes, please.
Given that there's a lot of geographic difference in the incidence of IBD, given the geographic variation in IBD incidence and Australia being rather a hotspot and everywhere else not, do you think the microbiome is playing into that? And if so, how would you control in your study for the fact that you're dealing with Australian microbiomes as opposed to American microbiomes or European?
Well, that's a good question.
And Sophie, just come back to the microphone.
That's indeed a good question, but I think as the pilot study, we could first focus on the Australian population, but then we could have collaborators from Europe or from the US to confirm these. Some studies have been done already, so I'm on the gut symptoms and the blood microbiome. So potentially we could also look into the literature and see the database that I've already produced, be able to compare if we have a geographic microbiome already in these cases, maybe.
So I said I wouldn't ask a question, but I will. So in terms of spondyloarthropathy, as you know, that's a multitude of diseases that comes under that umbrella. Are you after a particular disease subset, like ankylosing spondylitis versus psoriatic arthritis, or what are you after?
We are interested in psoriatic arthritis. At some point, we are interested also in the skin microbiome to try to link everything, but not all psoriatic arthritis patients have psoriasis, so it makes it difficult that way. But yeah, that's the disease we want to target first.
So you could have a collaborator that was a rheumatologist or a dermatologist, because those people would have psoriatic arthritis under them.
Yes, because those patients with psoriasis would be directed towards the SPA clinic if they get some arthritic symptoms coming in. Yeah.
Okay. Thanks,
Or
Anne-Sophie.
actually gastroenterologist.
Gastroenterologist, that's right. You've got a few to pick from. Thank you. All right, our next speaker is Dr. Debottom Sinha, as a research fellow at the UQDI, and going to talk to us on deciphering the functional role of MYC or MYC in driving resistance to therapies amongst HPV-driven malignancies. Thank you.
Thanks for the introduction. I'm a research fellow in the Fraser Laboratory. I just joined this year in January. I'm actually working on HPV-driven malignancies, and the picture here depicts what are the different type of malignancies that HPV drives. HPV, or people know it as human papilloma virus. You can see the numbers or the stats that has been shown here depicts how important this virus is with respect to driving tumorigenesis. And what I would like to show is the incident rates, especially oropharyngeal cancer, is actually rising pretty intensely in Australia. And not of like last year, it was reported that about close to 5,000 cases of oropharyngeal cancers in Australia. Importantly, about 70% of these oropharyngeal cancers are driven by HPV, so it's an important disease to be taken care of. The picture here depicts that what are the existing therapies which are currently available to treat these diseases in clinic. But I would like to again showcase the problems that the clinicians face, or overall depicted in the literature about these existing problems, which are resistance and metastasis based on the disease burden, tumor heterogeneity, which is actually fueled by tumor microenvironment, the immunoregulatory, the cellular components which are responsible for immune suppression, lack of antigen presentation, and poor trafficking of cytotoxic T cells to the site are very, very common problems. So what are the solutions? This is where I've been, like, my PhD comes into the picture, wherein I've been studying the molecular mechanisms driving tumor, but I've now started focusing on the fact that these genetic modulators or the regulators, can they actually be also involved in driving the impact of the tumor microenvironment? And one of the candidates that I've been studying during my PhD, also my first postdoc and moving into my second postdoc, is MYC, because MYC has been studied in the last 30 years for doing a lot of impacts that is associated with cancer. But in the last 5 years, MYC has been shown to highly regulate the tumor microenvironment, as is shown in the picture, that whenever the MYC is overexpressed in cancer, it does a lot of immunosuppressive functions, as indicated by the red suppressive arrow, which shows like how it stops immune surveillance and evades the immune system. So interesting fact that MYC, even though it's an interesting target and it is highly expressed in HPV-driven malignancies, it's not been explored in HPV treatment. And even though it's an interesting candidate, it cannot be targeted directly because it's a transcription factor. So in the industrial field, people have come up with inhibitors, which is BRD4 inhibitors, which are currently under clinical trial, and they have been shown to inhibit MYC transcriptionally. So with that particular idea, I like to pitch in my hypothesis wherein inhibition of MYC could potentially overcome hostile tumor microenvironment and boost clinical outcome of immunotherapies. My aim is to characterize the molecular changes observed in the microenvironment post inhibition of MYC with the methodologies involving syngenic and humanized HPV mouse models, which are currently available in the lab, with a downstream technique of spatial omics and flow cytometry. I don't need to give an introduction to spatial omics because Arutha has already given a brief introduction to that. But ultimate outcome that I would like to achieve is to have a strategic foundation for immunomolecular therapies, which could potentially boost clinical outcome in future. And what I'm looking for a potential collaboration is to get access to human PBMCs and biopsy samples of human tumors pre and post therapy. So yeah, that'll be good. With that, I'd like to thank you and happy to take questions.
Thank you. So the description of PBMCs and biopsies from human tumours is pretty broad.
Yeah.
Can you bring it down any further? HPV-driven tumours specifically, or?
Yeah, so, well, HPV patients, as you say, the number is pretty small. It will be great to have them, but also I would like to implement whatever the outcomes that we get from studying HPV into other cancers as well, so that we can have this type of therapies not only restricted to viral-driven cancers, but the idea overall is to repurpose existing drugs in combination of immunotherapy. So the reason being, we want to screen those PBMCs of patients of irrespective of the cancer type about the immunosuppressive molecules that is secreted in the plasma or the serum. So if a broader range of cancers patients have them, can the MIC be used in this particular scenario as well? So yeah, it's an open field.
Are you after samples that have a clinical characterization around them as well?
Yes, it'll be interesting to have something like post-chemo, because most of the patients who go into immunotherapy, that is especially anti-PD-1, have failed chemo. And the reason why anti-PD-1 or CTLA-4 are now being administered is because they exist in the market. So I would like to have much more tailored, improved therapies after understanding personalized needs. And that will be interesting to actually have more of a personalized or precision medicine approach.
Other questions? Oh, we'll take one from Derek here.
Hi, nice talk. Which MIC are you after? Is it CMIC or NMIC or IMIC?
So I would say overall MIC, because BRD4 kind of transcribes, it's transcriptionally regulate MIC. Now, I know there are different forms of MIC, but every form has different function. I would like to suppress all of them together and have a look into the overall impact. And BRD4 inhibitors have been shown to inhibit all the isoforms of MIC, so that's interesting.
I'll just carry on. So it's, is MARX also a target then? Because MIC binds to MARX before it is active.
Yes, that's another angle, because at pharmacological level, there are two different ways. Yes, one is BRD4 and one is by inhibiting MAX. That's another approach, but comparatively, looks like the BRD4 inhibitors are now in clinical trial phase 3. And even though none of them have been got approved, but they will be likely the best scenario to look into. But obviously, we can have a comparative analysis as well. Yeah.
One last question down here.
In terms of the clinical collaborators, do you have a preference in terms of what might be good to start with? Or have you, used particular samples before, which then might narrow down in terms of what sort of collaborators?
Yeah, I think the ideal way to start will be the HPV-driven oropharyngeal cancer. Okay. To start with, because that's where I'm specifically looking. It'll be nice to have pre and post chemotherapy or follow up after immunotherapy to see whether MIC is still upregulating. And if we can inhibit MIC in our mouse model in combination of, let's say, whatever the immune markers that is upregulated, can that be used for a better outcome in mice? That will be awesome to actually start with.
And what sort of sample sizes are you looking for?
That's again a statistical question,
but
anything above 50 will be a good start to start with, because HPV is not that easy to get in Australia. But obviously, international collaboration will be ideal. But yeah, let's see how it goes.
Thanks.
Thank you.
I'm very pleased to introduce our next speaker, another one of our clinicians from PA, Associate Professor Ross Francis from the Department of Nephrology, and he's going to talk to us on MR spec to predict progression in chronic kidney disease patients. Thanks, Ross.
Thanks, Helen, and thanks for the invite to talk today. So I'm going to present, I'm a kidney clinician. I'm going to present on behalf of my collaborators who are basic scientists and imaging experts. And we're looking at chronic kidney disease, which sadly is a common problem in Australia, estimated to affect 10% of the population, but often unrecognised. And as CKD progresses, this causes significant comorbidity. So apart from kidney failure, it's also a major risk factor for premature cardiovascular death. So it causes a lot of harm and kills about 65 Australians every day. But it's treatable, and if you catch this disease early, many patients, you can ameliorate their prognosis. We classify CKD based on measures of kidney function, so eGFR on a blood test, and we look at proteinuria, so albuminuria typically these days. And these are reasonably good at identifying patients with kidney disease, but they don't have great sensitivity for early disease, and their predictive value for the future is not fantastic. What we're looking at are novel magnetic resonance techniques to try and get more information about patients' kidneys. So MRI can give you information about the kidney size and anatomy, but nowadays you can also get information about fibrosis non-invasively using techniques such as diffusion-weighted imaging. But the real novelty that we can bring to this is the ability to actually estimate the concentration of metabolites non-invasively using MR spectroscopy. So we are just embarking on a clinical study called Discern CKD to try and ask whether these MR biomarkers can actually improve our prediction of patients' kidney prognosis. So we're just at the outset of this. We're trying to recruit 100 patients with CKD stage 3 to 5, as well as some healthy controls, which will probably come from the living kidney donor population. We have ethics and governance in place. We're ready to start recruiting. And we're aiming to scan these patients to look at their biochemical profile on MRS and then correlate this with their clinical data. So really, we've come into this looking to try and apply for some more grant funding to try and increase the number of patients that we can recruit. And if there's one thing that we really lack, it's high-level biostats collaborators, because this will generate quite a complex data set. But yes, if anyone is interested in collaborating in this project, we'd love to have a chat to you. So thanks very much.
Thanks, Ross. Questions?
The EU had a very big MRI and kidney collaboration that wound up last year. How does this align with the work that they did, and are there learnings from what they did and didn't achieve that could be applied here?
Yes, that's a great question. So there was a previous project very similar to this that looked at kidney cancer and kidney masses. It was possible to differentiate significantly between the different types of kidney tumour and identify malignant versus benign tumours with a high degree of precision using MR spectroscopy. So that's really where it led to this project, that we think this has significant promise for identifying patients who are highly likely to progress. And I guess with some of these new therapies that are coming out, like SGLT2 inhibitors, we may actually find that we can improve the metabolic profile in the kidneys. So this could eventually be a therapeutic target as well, we hope.
So Ross, there are biostatisticians and biostatisticians of various types, and in this building there would be a number of people working on various areas. What do you specifically need?
So we're going to end up with a large number of biomarkers, essentially, that we're going to have to correlate with clinical outcomes. So it's really about modelling and biomarker expertise using large data sets. That's really a skill set that we don't have at the moment in our collaboration. So that's where we're heading.
Yes.
I guess it's more of a, we might be able to help you. We've got a bioinformatics team, and they do imaging-based machine learning and AI to identify prognosis from these. So it might be linking up with them.
Yeah, that would be fantastic. I'd love to have a chat. So thank you.
We'll also talk about the at the end the CSIRO OTRI grants that are coming up, and potentially there could be some links there for you as well.
Yeah, we've been looking at those as well. Yeah.
Good. Okay. All right.
Thanks very much.
Okay, we are running to time, and I appreciate everybody who has presented so far in that regard. So our next presenter is Dr. Suganda Bhatia, who's a postdoctoral researcher for QUT, and to talk to us about targeting aneuploidies in cancers. Thank you.
Thank you to the LINC organizers, and I'm representing QUT and Pascal Digit's lab, where I'm working as a postdoc. So the TCGA, the Cancer Genome Atlas, actually covers a lot of cancer information, like including whole genome sequencing, methylome, as well as the RNA sequencing analysis. And it contains the data over more than 30 to 60 different cancer types, as well as for more than Okay, the link is not coming up, but yeah, so it contains the data for more than 30 to 60 different cancer types, as well as, so we all know that cancer is really very heterogeneous, but one of the things that is ubiquitous within the cancer is the aneuploidy, that is the deviation from the 46 chromosomes. And then this deviation appears because of the errors either within the DNA repair, replication errors, or because of the chromosomal segregation. So our lab in 2020 published this work where we actually identified using the TCGA data set that there's a lot of chromosomal arm aneuploidies, and it is present in more than 80% of the cancer, and it's actually significantly higher in metastatic patients. And also using machine learning and AI, we tried to predict the drug responses with regards to this chromosomal arm aneuploidies, and we actually identified various hits and the targets. And currently in the wet lab settings, we have just started validating these things. So there's a lot of chromosomal heterogeneity work that is being studied across the patient cohorts, and the significance of different chromosomes across different cancer types. And also that's why this translational research is very much important, because we all know that tumors are really chemo-resistant, or they are inherently chemo-resistant, or they become chemo-resistant. So targeting aneuploidy is an excellent strategy, because we know that they are highly prevalent in cancers. As I said, that more than 80% of the cancers are aneuploidy, and they are excellent predictors of the drug response. And also it has been found that with regards to whether if there is a large genomic changes, either its loss or gain of these like amplification or losses can actually result in poor prognosis or worse prognosis within the clinical cancer cohort patients. So, and then just last month, there has been 3 back-to-back Nature papers which have highlighted the whole pan-cancer compendium of this chromosomal instability or signatures for these chromosomal aneuploidy. And also the third paper highlights the functional consequences of like inhibiting, or when like why in the metastasis it's being higher and how we can inhibit it. So our aim here is right now what we are seeking from a clinical collaborator is a clinical expertise in cancer pathology, or who knows the treatment of the different cancers. Preferably our interest right now is with breast, ovarian, lung and melanoma. And also we wanted to facilitate this research in order to acquire the patient-derived cancer cells or the tissues. And also we would like to take their help in assisting the pathological annotations of the cancer, as well as in guided response to the drug therapies. And that's from my side. Thank you.
That was almost perfect, 301. Very, very well done. Questions? Well, you've got everyone this afternoon thinking. Here we go.
How far off do you think you are from actually coming up with a therapeutic that could go into a clinical trial?
I'm sorry.
How far off are you from coming up with a therapeutic that could go into a clinical trial?
I think because of the heterogeneity within the cancer, we have just started with the clinical, not the clinical trials, but the drugs that have come up with the chromosomal arm aneuploidy scores. So we are right now currently validating that within the cancer cell line settings, or also the biobanks that's present with Mater, with the breast cancer, as well as we are trying to connect with other clinicians. But with the LINC, we actually are trying to seek like a long-term collaboration that still can be ongoing, and with the patient data sets that we can still hope and validate. But I know every cancer is heterogeneous, and so it really has to first undergo the whole genome sequencing part in order to really analyze how many copy number changes are there within that cancer.
And the drugs you're looking at, are you
so
repurposing?
Same like the bottom, it will be a tailored, like a precision medicine approach.
And yet, so they're drugs that are already
like if there's
approved.
a specific chromosomal arm amplification, let's say corresponding to MEK upregulation, or so it will be specific precision therapy targeted to that. Yeah.
Excellent, thank you.
Thank you.
Just bear with us for a moment, because our next speaker is going to be online, and it's Dr. Gishan Ratanaki, who's a radiation oncologist from the PA hospital. And thanks for talking on the development of novel radiation drug combinations. Go for it, thanks.
Thank you. Thanks for this opportunity to speak. I'm going to be talking about combining radiotherapy with some of the novel cancer therapies being developed in Brisbane today. So radiotherapy forms one of the cornerstones of cancer management, and it's usually delivered in the form of high-energy X-rays with a machine such as this, a linear accelerator, and it works by causing DNA damage to the cancer cell. About 50% of cancer patients receive radiotherapy at some point in their treatment journey, and about 60% of the time it's used with curative intent. Because such a large number of cancer patients receive radiotherapy, and it's used often with cure in mind, it means that any improvement in the efficacy of radiation really has a very significant clinical impact. So the idea of radiotherapy is to do maximum damage to the tumor while sparing the normal tissue. So in terms of improving the efficacy of radiation, there's two main ways. The first is with technological innovation. So this is where we try to increase the precision of the radiation, and there has been a tremendous progress that has been made in that field over the past 20 years. We can deliver higher doses to the tumor than we ever could before while sparing the normal tissue. This is really translated to better tumor control and lower the normal tissue toxicity. The second way is with radiobiological innovation. This is, for example, using drugs to sensitize the tumor to radiation. And the last great advance in this area has been the introduction of chemotherapy with radiation. And chemoradiation has become the standard of care in many different cancer types. But there hasn't really been too much gain since then. And this is despite in the area of systemic therapy, there being a revolution with the introduction of targeted therapies and immunotherapies. We are yet to see these new generation therapies being used constructively with radiation in the clinic. So we think that radiobiological innovation might be the next frontier in improving the efficacy of radiation. And we're interested in taking some of these promising biological agents that are either approved for use or are in the pipeline and finding out ways to study them in combination with radiation in clinical trials. There's many different issues that are preventing this, but one of them is the lack of preclinical studies and good preclinical data. Either the studies are not done or they're done poorly. So what we want to do is we want to take some of the cancer therapies that are being developed in Brisbane and we want to test them in combination with radiation in a rigorous and systematic fashion. And this is really going to be helped a lot with the installation of the small animal radiation research platform at the TRI, I think in the next month or two. And this piece of equipment allows us to very precisely irradiate tumors in mice using a lot of the techniques that we would use in a clinical machine. So if you're a research group that has or is developing cancer therapies that might have some promising combination with radiation, then we're very excited to talk to you. Thank you.
Excellent. Thank you, Gishan. Questions? I guess if you have preclinical models in mind in terms of already established preclinical models for particular cancers, or what's your preference, or what do you need in that preclinical collaborator?
So I think the simplest model is a sort of flank xenograft, and as a patient-derived sort of xenografts on the breast, but you're just using tumor cell lines is fine. The advantages of this machine is that it images the mouse, and therefore we can treat orthotopic sort of tumors as well. So, but I think to begin with, a quite simplistic sort of flank xenograft would be more than adequate.
Okay. Other questions? Rick, please.
Yeah, thanks, Gishan. The nice presentation. And so I'm just wondering, when you mention the chemo radiation, does the chemo actually sensitize radiation, or they just combine to work? You're using the chemo to kill the cells as well.
Yes, so there is a synergism, for example, with platinum and radiation. So you get better local control with the addition of chemotherapy, but you also get better distant control as well. So the chemotherapy obviously has an effect on outside the radiation field on microscopic disease. So there is it goes both ways.
And a second question, if there's time, would be what would be the sort of endpoints would you be looking to see a better size reduction in the tumor, or a lower dose of radiation required, etc.?
Yeah, I think both. I think we would like to improve the tumor control and therefore improve survival. But I think de-escalation of radiation dose, which we know would improve the normal tissue toxicity, would be very welcome as well. And for example, in HPV related cancers, such as in oropharyngeal cancers, there's been a real push to try to de-escalate radiation dose with the use of systemic therapies.
Thank you. Thanks for coming online today, Gishan.
Thank you very much.
So our next speaker is Dr. Marcus Gray, who's a senior fellow at the PA Hospital and with UQ, and he's going to talk to us on social reward learning in schizophrenia, computational modelling with variational Bayes. Thanks, Marcus.
Thank you for the opportunity to present. This is going to be quite a change of pace for most of you, I think. This project is essentially a neuropsychiatry, computational psychiatry project, so much less cellular immunology and much more working with psychiatric disorders and trying to improve the mind. So our project is about social reward learning in schizophrenia. Schizophrenia, as you all know, is a relatively prevalent disease, approaching about 1% of the population at any one point in time, and it's got quite a complex symptom profile, but overall, very generally, there's negative symptoms, which can be typically, will include things like avolition, affective flattening, and anhedonia. And it has a devastating burden of disease that often is lifelong. One of the features of people with schizophrenia is they have poor social relationships, and this in itself can really exacerbate the disease burden. So they're much less able to form good social relationships, and that makes their lives much harder. So that's the focus of this project. We're looking at a group of clinical patients through treatment, and we're focusing on social processing and in particular social reward learning. So why do we think that might be interesting? Well, in these patients, we know that learning generally is compromised. They don't learn as well as people without schizophrenia. And we know that primarily through experiments with reinforcement learning or reward learning paradigms. These are very sort of simple experimental tasks which you give people, very simple learning exercises, and with that you can demonstrate sensitivity to reward, punishment feedback, and how that underlies most of the learning that we all do naturally every day. So we know that there is a deficit in reinforcement learning in schizophrenia, but the question is there a more, within that, is there a more specific additional burden in social reward learning? So, that's our first major question of interest. And secondly, we're also interested in whether a therapeutic program of cognitive remediation, which specifically focuses on social cue processing, can this help improve this deficit? The approach we're going to take is to apply variational Bayes to this framework. It's quite a novel mathematical framework, which I think is really well suited. And I just wanted to basically explain that picture on the left there sort of explains the overall Bayesian framework. Essentially, you can set up a computational model where you simply give someone a learning task, you give them inputs, which is the reward before each of their choices, and the other thing you provided are the observations, so you watch what behaviours they make. And from that, you can construct a variational Bayes model to identify what are called the hidden states of the model. That essentially is the value that people place on certain behaviours. This is an example, will seem very simple, but this is the experimental task we're using. We ask people to try and please this person by making one of 3 button presses, one of these 3 button shapes. So they don't know what the result of any button press will be until they trial and error. So they might start pushing one button and find that that has a good effect some of the time, and they might explore and find out eventually that she seems to like one of these buttons, and through trial and error, they will work out an optimal response. Essentially, they're learning the hidden states for each of those button responses, but the value of each response evolves over time. It's a dynamic task, so they have to keep learning, keep updating their calculation of value of each of these responses. It's a very simple task, but that allows us to computationally model the acquisition of learning. And this is the social learning paradigm because we're using social feedback. We have a similar non-social task which we're comparing with it. So that's very simply, that's the basis of it. If we apply this variational Bayes framework to some real data we've collected, we can calculate out the hidden states for each of those choices on a time-sensitive manner. So at each second, we're able to update the value of each response. We can then use that to generate a prediction for social reward learning, and we know there's dopaminergic systems in the basal ganglia, which are very good with reward prediction error, and this is the neural circuitry which underlies this sort of learning on a real-time basis. So we want to get patients and give them this learning task, find out how they're doing, find out if there's a specific social reward learning deficit, and then look at the effect of cognitive remediation therapy on their behaviour and on their neural function during these learning
Marcus,
tasks.
I promise to be strict on time. Tell us about your collaboration requirement.
Okay, so essentially we've got a good team. We're building a clinical neuroimaging research network with a number of different patient groups. But for this project specifically, we need a bit more expertise in variational Bayes. It's hard to come by, and we need some extra funding to carry the project forward.
Okay, questions? I think it's the mathematical modelling that put them over the edge, Marcus.
It's very different from probably every other presentation, so
No, here we've got one down the front here.
In terms of the mathematical modelling, so the Bayesian models, do you require research groups that have had specific experience in these neural applications before, or are you okay with talking with groups that are just generally great at Bayesian models? How much specific expertise do you require?
We can get by without finding a group that is doing this specific thing.
Yep.
But there's one main modelling group in Paris that I'm very keen to re-establish connection with. But I think generally people who are used to thinking in Bayesian terms would be very useful just to throw around these ideas with.
Okay, thanks.
Okay, thank you, Marcus.
Thanks.
That's fine. So our next speaker is Chimakara Layanje, who's a PhD candidate in the Bachelor Lab with QUT, to speak on the long and short of long non-coding RNAs in ovarian cancer. Thank you.
Thank you so much for the opportunity. And I'm Chimakara Layanje, finally a PhD student from the Molecular Genetics Lab, QUT. And this story is about the most lethal forms of gynecological diseases among Australian women, and that is ovarian cancer. More than 1,500 patients are being diagnosed each year, and more than 1,000 women die because of ovarian cancer each year, but still there's no effective diagnostic test available for this disease. Regardless of the tissue and genetic diversity, most of these patients are being treated similarly over the past 3 decades. And most of the patients develop treatment resistance. Actually, 80% of patients will develop treatment resistance, and they grow into advanced stages with less than 30% of survival. Unlike other cancers, ovarian cancer does not quite get the same attention for research funding, but researchers are still trying to figure out what is this disease and what are the causes of the disease and how to develop effective clinical treatments. And answering these questions, we looked deep into ovarian cancer tumors, where we discovered a novel microprotein, a novel group of microprotein that might answer the question of how to cure ovarian cancer treatment resistance. And microproteins have been previously ignored by researchers because of their small size. Surprisingly, these microproteins are produced by genes which previously believed as non-coding genes that do not make any type of proteins. Apart from the discovery of this microprotein, we observed the microprotein levels are being reduced in response to the chemotherapy resistance. That suggests that this microprotein might have an anti-tumor role in inhibiting ovarian cancer progression. To confirm this biological role, next we intend to overproduce these microprotein genes in ovarian cancer cells and animal models by using a viral vector-based gene delivery method. By using this preclinical evidence, we expect to introduce the first ever microprotein gene therapy for any of the cancers in Australia, and it will be for ovarian cancers for the women diagnosed with ovarian cancer actually. Because of the high action potency of microproteins, their anti-tumor activities will be highly reproducible, and these viral vectors, viral delivery vectors, can be further modified to improve their stability, safety, and biocompatibility with human cells. And not only that, we will be introducing a novel diagnostic or a prognostic test that will be accurately quantifying these microprotein inside clinical tissues that can, of course, will be an accurate test for diagnosing and prognosing ovarian cancer in clinical stages. Also, we have tested this concept for prostate cancer as well using NHMRC and Cure Cancer funding. And we have observed a different micropeptide that is critically involved with prostate cancer progression. And we would like to collaborate with any of the gynecologists and endocrinologists, and we would like to share your clinical expertise with us, and we would like to access clinical samples for ovarian cancer, particularly aesthetic samples and serum samples, and we can use them for our validation of our own research findings. And we believe discovery of microproteins is not just it won't change the legacy in conventional cancer treatments. It will open up new avenues in any other modern medical sciences. But nevertheless, microproteins are still considered as Lilliputs in biomedical research, but never underestimate little things, because it will be the little things that are going to make big things happen. Thank you.
Excellent, thank you. Questions? So not my area, obviously, but if ovarian cancer, predominantly women do very poorly once there is chemo resistance or otherwise. So to work out who the viral vector, group, how do you stratify ovarian cancer when predominantly everybody does so poorly?
In terms of the microprotein gene therapy? Every gene therapy comes with the combination therapy. We won't use that as in standalone therapy anyway. So gene therapy has been tried for ovarian cancer before, like P10, P16 and P53, along with the combination therapy with platinum-based chemotherapy for ovarian cancer. And we believe introducing gene therapy will reduce the chemotherapy use, like the dose usage, and it will reduce the side effects that's along associated with the chemotherapy resistance as well. And it will reduce down the number of cycles as well as number of period, like 3 or 6 months, the chemotherapy treatment. So it will reduce the cost and the tumor burden as well. That's the whole idea of using gene therapy.
Okay. Other questions? Yes, please.
Do you have any idea what the interaction partners are for the microprotein?
So still we haven't done any functional assays for this particular microprotein. So we'll be using overexpression models for this particular microprotein, and we'll be looking on, so this microprotein is produced by a London coding RNA. So we want to see whether this London coding RNA has a function independently out of the microprotein, or microprotein has a function independent out of the London coding RNA as well. So we'll be looking on 2 different pathways to figure out what are the interacting partners, what is the subcellular location of this particular microprotein and the London coding RNA, and how they interfere with the tumor growth and the metastasis. Still we don't know. With the overexpression models, I think that's why we are looking for money and to grant support just to go forward with the functional analysis.
Yes, from a drug development perspective, understanding what your on-target toxicity might be in cells other than tumours would be quite important as you go forward.
Just another question up here.
Has this gene therapy been successfully established in diagnosing or reducing the effects of cancer in any of the cancers?
You mean microprotein gene therapy?
Yeah.
Yeah, microprotein gene therapy hasn't been introduced to anywhere in the world, but there's one preclinical study is going on microprotein gene therapy for cardiomyopathy. And it's been validated that this particular microprotein called as dwarf been successfully reduced the cardiomyopathy burden and muscular distress. And it hasn't been tested for any other cancers, but there are, like this particular field is very new for cancer. I mean, around 20 to 25 microproteins have been properly validated for their biological function in cancer, but none of them has been to clinical phases yet. But there are other viral vector gene therapies for ovarian cancer as well as in other cancers. Yeah.
Thank you very much.
Thank you so much.
Now, lucky last is Dr. Karina Walpole, who's a research officer with Bada Research, to talk to us about humanised mouse models from research to the clinic, a solution to bridging the translational gap. Thanks, Karina.
So firstly, I'd also like to thank you for the opportunity to talk today. I am Karina Walpole and I manage the humanised mouse facility that is an outgrowth from Kristen Radford's research group here at the Bada. So humanised mouse models are a unique model that is created by using an immunocompromised mouse, which we engraft with human stem cells. And this allows for a human immune system to grow and develop inside these mice. So throughout my research career so far, I've seen so many fantastic ideas and research and novel therapies and drugs that are coming through. But the reality is that only about 85% of these actually make it through from research into the clinic. And this is where humanized mice can come in. They have the ability to bridge this translational gap that between our basic research and into the clinic. So an example of this is with the immunotherapy theralizumab. So this immunotherapy was developed in the lab. It was shown to be safe in rats and in non-human primates, and it progressed to clinical trial. And unfortunately, in the phase 1 clinical trial of this drug, all of the healthy volunteers became critically ill, and it was terminated. And later on, this immunotherapy was later able to be tested in a humanized mouse study. And the similar results were found in the humanized mouse study, showing that these mice are able to work as a warning system of what will actually happen in people. So in our group, we have been uniquely positioned to be able to actually develop these models and optimize them. And this is through our long-standing collaborations with the Queensland Cord Blood Bank to get the cord cells needed to create these mice, as well as our fact that we are here at TRI and we have access to the state-of-the-art facilities that are here at TRI. And so what we have done is as part of our group, as we've developed the humanized mouse facility, which is a platform in which we will be able to share our expertise and understanding and knowledge of these unique models with other researchers collaboratively. So specifically, the applications that we've been using these mice for our own research is in tumour modelling and development of immunotherapies. However, they can be taken forward into many other options. We've collaborated with people in infectious disease so far, and they can also be used for safety and efficacy testing of novel therapies. So what we are looking forward for in a collaborator is to find people, both clinicians and researchers alike, who would like to develop models that have an immune content that we could be adapted into these humanized mouse models. Or we're also looking for people who have got new therapies or drug ideas that they would like to test in the context of a functioning human immune system. Thank you.
Excellent. Thank you, Karina. Questions? Yes, please.
Hi, can I ask, a lot of animals proceed for, do you ever see GHD in a model or might help?
Yep, so the context of the time at which these models can be used is dependent on the base strain of the model that we're actually looking at and what you want to be doing. So most of our models, they develop the immune system somewhere between 8 to 12 weeks. Some of our models are useful for another 6 to 8 weeks after that. Other models are quite happy to use for much longer periods of time. The models we do see in these mice, so we don't see GVHD in the sense that you have a straight up in these mice. However, what we do see sometimes is at the very long term, at the end of the useful period of these mice, then you will start seeing some GVHD-like systems. But the fact that with our models, in the way we create them, those symptoms are a lot less than some other models where you're using straight PBMCs and putting them straight into mice. The fact that we are building them from stem cells and putting them into the immunocompromised mouse strains, that is less of a problem. You have an area where that isn't. You can usefully use them.
Thank you. Other questions? Yes.
So they look like a really good model for toxicology. I have two questions. One is, will the TGFDA recognise them as models? And is there any plans to do the lab to become a GLP lab?
Longer term, yes. Longer term, we really want to sort of build this model, build the capabilities facility to actually expand it further. At the moment, we are just looking for collaborators to develop the models and develop, research projects, but that's definitely a long-term thing that can be looked at, yeah.
Thanks.
Thank you, Karina. Excellent. We have come to the end, and I have found, as I did last year, the LINC grant talks to be both informative as well as to make us think about what we might work on moving forward and who we might work with. So on that note, the collaborations, all of the things that we care about, which is the money. The grant process will open as per the dates. You need to find collaborators that are early to mid-career, that are a clinician and researcher coming together. That's a new collaboration. And the PIs, who are the early and mid-career researchers, need to have significant mentors, a clinician mentor and a research mentor. And these collaborations are for $50,000 for coming together for a period of 2 years. I also remind you that we have the CSIRO and TRI grants. And again, this is a collaborative opportunity that is not tied to career stage. And it is about researchers coming together with clinicians and coming together with the Australian eHealth Research Centre. And they are $100,000 grants, and they'll be opening on the 8th of August. If you have any queries about the grants themselves, about collaborators, about navigating the difficulty between coming together with a clinician or a researcher, I encourage you to contact Benny Watson, who is here at TRI and is there to help you with those connections, or any of the other TRI staff, including myself. And that just leaves me to thank you all for coming and stay well. Thank you.